All right, we are back from the 2025 Borealis Psychedelic Science Summit in Stockholm. My name is Yoshi, and I'm joined now by Professor Suresh Muthukamaraswamy. Welcome, Professor.
Hi, Yoshi. Nice to be here.
Nice to have you here. How are you doing?
Yeah, good. It's been a long but really enjoyable conference.
Yeah.
Well, I've flown from New Zealand, so
Wow!
Yeah.
All the way from New Zealand.
Yeah.
How many days ago did you get here?
I got here 2 days ago. Spent a day at Uppsala, but I'm in Europe for 3 weeks, so I didn't just come.
Okay,
this.
good.
This is the start of a little.
How long was the flight?
It takes about a day, right?
Takes about a day. Yeah.
17 hours from Auckland to Dubai and then into Stockholm. Yeah.
Oh, right. You have to go to Australia first before going anywhere.
You can go Auckland, New Zealand.
Sorry.
Auckland, New Zealand.
For some reason,
Dubai.
I thought of Auckland being in Australia.
Crimes.
It's been a couple of long days for me as well.
Yeah, I bet. I've seen your schedule.
Yeah, you're actually the last interview I have. So 21 out of 21. It's been a busy couple of days.
Yeah, congrats to you.
Very fun though. Yeah. Very fun. Yeah, I got the luxury of having a in-person run through of everyone's presentation, which is super cool. So happy about that. So tell me a little bit about your background and how you got to working in the psychedelic field.
Yeah, my background is in general psychopharmacology and neuroscience. So I was really doing neuroimaging during my PhD and my first years of my postdoctoral fellowship. I always had an interest in pharmacology, but I was more, as I was doing imaging, I became interested in like how you can manipulate those signals. So then I started doing general pharmacology to try and work out how we can change those kind of EEG or fMRI type signals using pharmacological interventions. So I've worked with a lot of different pharmaceutical interventions in the last 15, 20 years.
Right.
And then at some point in that study, I was doing a lot of work with anti-epileptics and anesthetics. And then around 2010 or something like that, Robin Carhart-Harris's team were coming into my center and they were doing their first psilocybin studies around then. And I was kind of looking at it, I was kind of curious.
Yeah.
It's like, they're probably not going to make that work. And they managed to pull it off. And then we were like, they had found some manipulations in the fMRI signal. And I was working in an MEG center at the time. And I was like, why don't we try and do your psilocybin study in my scanner? And that's how it started. So I did psilocybin imaging, and then we did LSD neuroimaging.
Right.
And then I was doing my own ketamine work because I was interested in the neurophysiology of ketamine. And then when I moved to New Zealand, I switched for funding reasons. I was doing this ketamine work, but then I started doing clinical work because that's what I could fund in New Zealand, because New Zealand's not interested in like basic research really.
Yeah.
So to fund it, so then I've been running clinical trials the last 10 years on ketamine and now LSD microdosing, amongst other things.
So interesting.
Yeah.
When you say manipulating the signal, what does that mean exactly?
Yeah, so because essentially, so if you put someone in a scanner, maybe they're not doing something or maybe they're doing a task, and you record some kind of signal coming out, right?
Mm-hmm.
But then you're like, well, what is the neurophysiological or neuropharmacological basis of that signal?
Mm-hmm.
And so to manipulate that signal, one way to do it is to put drugs into the person, right? And so maybe you record an EEG alpha wave.
Right.
And you say, well, what are the kind of physical pharmacological contributors to that? And if you manipulate, for example, the anesthetic, the GABA, then you can see what the effect of GABA on the EEG is, or in the case of the psychedelic, what's the effect of the serotonin 2A manipulation on your EEG and fMRI signal. So I have a very strong methodological background.
Right.
And more recently, I've been trying to apply that to the way we run clinical trials.
That's fascinating. So what kind of clinical trials have you been doing?
Yeah, so most recently, or over the grand scheme of things, we did a lot of work with ketamine early on. Because when I moved back to New Zealand, like I was saying, we had to do clinical work, so I could no longer just do the basic imaging and help and sort of more nerdy science stuff. So we were doing ketamine and depression, but our ketamine and depression studies, we were putting people in scanners and trying to measure, because we knew we would get an antidepressant response, and we were seeing if we could work out the physiological basis for that rapid antidepressant response. So we ran clinical trials with ketamine. We ran clinical trials with a drug called scopolamine, which is an antimuscarinic drug at very high doses. It's psychedelic, but at the doses we were giving, it was sub-psychedelic. We did transcranial magnetic stimulation. And then most recently, we've been finishing off a LSD microdosing trial in depression.
Interesting.
So yeah, those are the psychedelic-y type trials.
Right.
So there are depression trials. And then we've done a number of LSD microdosing trials. Our group we're quite a small country, right? And we have limited funding. So my kind of thing is to try and find a niche where we can do something that's like unique-ish.
Right.
And I found a niche over the last 5 years, and that was in microdosing, because I found something in New Zealand regulations where it was actually legal for us to actually prescribe in a clinical trial microdoses for people to take home with them. So it's kind of a quirk of the way our legislation is written,
right?
But there's nothing to restrict, say, something. If you want to do microdosing in other countries, you have to do it in the laboratory. Right. And because the substances aren't allowed out of the lab. Right. But in our regulatory system, I discovered that actually there's nothing to stop us.
Okay, interesting.
And so around 2019, 2020, I applied for permission to run these home microdosing trials where people would be in a randomized controlled trial, but we would prescribe the microdoses for them to take at home. Right. Like recreational or lifestyle, whatever, users do. So we had the ability to do that. So we've been focused a lot on that space because we're the only group that can really do it because of our legal system.
Interesting.
So
how many microdoses do people get to bring home at a time?
Yeah. So they're allowed to take about 5 doses home with them. Okay. So they're on trial for 6 to 8 weeks. Right. And they do about 2 a week or whatever. The first trial was every 3rd day, so they're doing 14 to 16 in total. Yeah. But we only give them 5 at a time, so they can't take them all at once and get a macrodose.
That's what I was going to ask. How many times do you encounter that someone runs out of them?
That has never actually happened. That's called stacking, where people and it's a theoretical possibility, right? But the reality is that in a clinic, people think, oh, everyone's just going to take it at once. Yeah. But if you think about actually from the participant's perspective, why are they doing the trial? Yeah. I mean, if they want to get, I won't swear, if they want to get LSD, they will just go down the street and get some LSD. It's not hard to find. You just go download an app, go on the black web and you'll get LSD, no problem. Like it's no hard to do. Yeah. People in healthy control studies, they actually want to make a contribution to science. Of course. And they're doing all the screening and they're doing all the screening and baseline testing. Man, there are a lot easier ways to get LSD than doing all that work just to get a few microdoses. In reality, it just doesn't happen. Got it. Yeah, so it's a theoretical possibility, but in practice it never happens. And I say that from having had now almost 200 people run through our microdosing trials.
I'm glad to hear that.
Never had a problem with that.
Gives me renewed hope for humanity.
Well, what would happen in a situation where maybe outside of a clinical trial, where there's a lower bar to entry, then maybe that would happen. But in our case, it hasn't happened. And if you were doing psilocybin microdosing, man, you can just go pick some mushrooms in New Zealand. It's not hard. There's easier ways than going in the clinical trial. For sure. Yeah.
So do you notice that there's manipulation in these signals even from a microdose then?
Well, yeah, it's actually really hard to. We found behavioral changes and where you can measure. So we do very careful placebo-controlled trials, and we do manage to find changes in people's mood and their feelings of creativity. And so there's definitely they feel happier and more creative and all that sort of stuff. But actually trying to measure brain signal changes at those kind of 10 microgram doses, honestly, didn't come up very successfully. Because we were doing EEGs and all that sort of stuff. That was actually not really very successful trying to find change. I think you need to be a bit higher in the dosing to more like 20-ish to get a clear signal. Right. There's other studies have done that with EEGs. Other lab-based studies with microdoses have shown around 20 micrograms that you get EEG changes. We were a little bit lower than that. But again, that wasn't our main, niche of interest. Ours was that actually we could get people doing 16 weeks of microdosing.
Right.
But what we didn't find was any change in like overall mood. So these were healthy volunteers. We didn't see that their like creativity actually improved, or their cognition improved, or their overall well-being improved. They had a really good day when they were microdosing. They enjoyed the days of microdosing, but there were no long-term changes that we could see in healthy volunteers. The depression data is still to be
right.
That's coming still.
Yeah. Interesting. What are you most excited about in your current work?
Current work, well, we are doing what I believe is we've become quite interested in women's health. It's really underexplored area of psychedelics, really neglected. Women, and that's just it's a symptom of a greater neglect of women's health, and particularly women's mental health. So we have just started what we believe will be the first menstrual cycle related psychedelic study. So what we were interested in is whether the response to, in this case, it'll be psilocybin, changes across the menstrual cycle. So what we're going to have is healthy women volunteers, healthy menstruating women will come in at 3 points during just after menstruation, during ovulation, and then just before menstruation. So follicular, ovulatory, and luteal. So the technical words. And we want to see if the response to psilocybin changes during those periods. Now, that might sound crazy, but it's actually not, because what we know from animal evidence is if you inject like rats with estrogen, the expression of the 2A, serotonin 2A receptor, the main psychedelic receptor, it actually like more than doubles, 200, 300%. Yeah. So the 2A receptor expression can be quite, at least in the animal models, can be quite sensitive to estrogen levels.
Wow.
So it's not unreasonable to think that maybe
not at all.
That it might change, but we don't know. So we are going to formally test it. Right. So I'm excited to do that study because it's really... so it'll be the kind of first thing to do. And then after that, we'd be interested in studying some of the women's mental health disorders, but that's in the future for us. We've got to go step at a time. So it's kind of fun though That's
super interesting. So if that in these mice where they double the amount of 2A receptors, was it the amount or?
Yeah, it's the density of receptors.
What could that mean? Like, would psilocybin give them double?
Well, maybe not double, but like you might get
an effect.
You might have more of an effect at certain stages of the menstrual cycle, and that would be important to know, right? It's important for
yes, that would be.
It's important for it's beyond psychedelics. Right. We know that across the menstrual cycle, women have differential responses to drugs, and this is largely neglected by the medical community. And some innovative physicians, for example, do dose adjustment at points of the menstrual cycle,
right,
where they, and sensitivity goes up. And there's all sorts of menstrual cycle related disorders. Like we're interested particularly in premenstrual dysphoria, but there's also things like menstrual related migraines. A lot of women can get what's called catamenial epilepsy, where epilepsy increases and changes, seizure frequency changes, particularly in the late luteal phase. So there's a lot of menstrual cycle-related physiology that is interesting to study, and potentially psychedelics may interact with that system and may have a therapeutic role potentially as well that could be explored. But, we don't spend enough time on women's health. It's a terrible situation.
Yeah.
If men had these disorders, they would be it would be front of mind, right?
Yeah.
If men suffered from this stuff, this stuff would be studied. It's just terrible for health equity.
So. No, that has been a recurring theme in many of these conversations that I have over the days.
Yeah.
It's just an underserved area, and there are all these excuses like it's more complex to study. Well, but
it is more complex to study.
We have to do it.
Yeah, it is more complex to study.
But if you say that, then you can't say take the same drugs, take the same amount. That's
it's more complex. Women's health doesn't get enough funding. There's there's other people talking about it. And luckily, we're the first to get started. Well, lucky in terms of the competitive edge, but not lucky for women.
Yeah, of course.
And we're really glad to do it. And my senior, she's now going to be a lecturer, Rachel, has been doing a lot of menstrual cycle work outside of psychedelics. So with the ketamine or epilepsy, and has done a lot of study of the menstrual cycle. Menstrual cycle studies are actually very difficult to do, to get the timing right, and like the management of participants, even healthy ones, let alone ones who have disorders. So she's got a lot of experience in that, so we feel pretty confident that we can do a good job of it after her now almost 10 years of experience doing menstrual cycle studies.
Amazing.
We're pretty confident that we can, like, with my expertise in psychedelics and her expertise in menstrual cycle research, so we're pretty confident we can do a good job of those studies. So
I'm sure you will. That's great.
Hopefully, because we never like to do bad studies.
Of course.
I mean, and we try to step through carefully.
That's great. Is this what your presentation was about during this summit?
No, not at all, actually. The other part of while you're running trials, long trials, there's like teams working away at stuff, and you have a lot of time to do stuff. And so one of my little kind of background hobbies was thinking about clinical trial design. And so, because that's something you can do while data's coming in. And so I've done quite a lot of writing on psychedelic clinical trial design around the problems of, like, blinding becomes an issue in psychedelic clinical trials, because patients know that they've had the intervention, and typically in a randomized controlled trial in medicine, we want the patients not to know what they've had. But in psychedelics, that becomes a bit of an issue because they know. And that potentially confounds the results. There's been a lot of talk in the conference about that. And so I wrote one of the, not to blow my own horn, but I wrote a quite influential article in 2021 about the blinding issues. And I've written a sequence of articles about that. And so we're just about to publish the new one. It's just going to come out any day now. And so I was talking about that paper, which is a new way to kind of a different type of clinical trial design that psychedelic science could use. So
can you give us a little preview?
Yeah, well, absolutely. Yeah, this one's not a secret. So normally when we run clinical trials, we run them quite in an abstract way. These are called explanatory trials, where we're trying to demonstrate that the treatment works. But actually the way we deliver the treatment is not at all like the real world. They're kind of in these highly selected patients that are not like what the patients are going to be treated. And the interventions can be quite contrived, like not at all actually like how it'll be delivered in the real world. So the patients aren't the same. The clinicians do it a bit differently. We make the patients do all these like weird outcome measurements and interviews that never would happen in the real world. And so the actual trials themselves, nothing like how treatment would happen in reality. Now, there's, outside of psychedelics in general medicine, there's a class of trials called pragmatic trials. And those pragmatic trials have been developed to basically answer those questions. Well, actually, how effective is this treatment in the real world? And so you can design clinical trials using pragmatic trial principles to basically answer that question. It's like, how actually effective would our treatment be if we actually deliver it like it would be delivered in real care? How effective would it actually look? So my talk was basically not particularly genius, to be honest. It's just like, well, if we were to do pragmatic trials in psychedelics, what would it look like? And making an argument that actually we should probably be doing pragmatic trials in psychedelics, not to replace the normal kind of nerdy, weird trials that we're doing, but that pragmatic trials would be a really good complement to the current trials that we're doing, so that we would not only know that the psychedelics work, but we would know how effective they actually were in the real world. And why that's important is because, like, someone's got to pay for this, right? Like, and the people who are paying for psychedelic therapy, whoever it is, like governments or insurance systems, whatever, individuals, they need to know how effective it is, right? Like, is this worth spending $30,000 a patient for? Is it cost effective? And in order to do that, you actually have to have data on how effective the treatment is in the real world. So the argument that we're trying to make is actually we should be shifting, like, let's let industry do the kind of weird trials to show that their products work, and academics and all the other parts of the psychedelic ecosystem could be doing more pragmatic trials to actually work out how cost effective it is or optimize the treatments for real world care.
Right.
So that was what my talk was about.
Interesting.
Yeah, maybe.
For sure.
Yeah.
Looking at the broader psychedelic research field, is there something you're particularly excited about that's going on right now or moving forward? Anything you would like to highlight that you've seen?
Yeah, I do think, over the years, things have changed. I think we have to be adaptable because industry has kind of taken over from where we were 5 years ago. Industry has come in and are running these big trials now.
Right.
And they have money and resources to run massive trials.
Right.
And this is part of the pragmatic trials thing as well, is actually academics can't do that. They run 400 people in a trial and we can only put 40 or whatever in. So we have to think a bit differently about what psychedelics could potentially, how we could be doing things. So thinking about innovative ways to use them. But I was interested to hear some of the other speakers talking about things like, prevention, for example, and just other ways that you could potentially look at how they could be used in terms of well-being and all those kind of things. So I think that would be kind of interesting to look at in the future, moving beyond just these really, really difficult pathologies.
Yeah.
It's pretty grueling work, and I still think there's a lot of really interesting things in psychedelics that could be looked at beyond just treatment-resistant patients.
Yeah, of course.
There's probably still a lot of really interesting questions. So a few things that got me thinking. The question is how easy it'll be to fund those kind of studies,
right.
and how to do them. So
yeah.
And I think the other thing that I've been thinking about lately, and it's also not necessarily related to psychedelics, is we've spent a lot of time worrying about pharmacology, 2A receptors, 2A, 1A ratios, what the pharmacokinetics are, and that's all kind of nerdy pharmacology stuff. But I have this kind of intuition that actually, and it's not just intuition, there's plenty of kind of data, anecdotal data to support that actually, it's probably bigger influences on the psychedelic experience are actually people's what they know, the social and cultural context of how they're taking the drug, environmental factors that might manipulate or kind of influence the psychedelic experience that they have is probably is heavily influenced by, cultural and environmental social factors more than just the 2A, 1A pharmacokinetics. Because most people can't tell shit about the drugs they take, right? The classic one is cannabis, right? So if you give, going well off topic, but people talk about cannabis and entourage effects and CBD and stuff, but if you run double-blind trials, man, no one can tell the difference. If you give them 15% THC with CBD, without all the thing, no one can tell the difference. It's much more influenced by people's perceptions.
Right.
I remember once when I was a student, I was like, man, I love Coke. So we did the double-blind test and I had Fanta and Coke, and I couldn't even tell the difference, right? Close my eyes and it's I think that one's the Coke. No, it's the Fanta.
Wow.
It's so much of which is in our head, right? So I think that there's a lot of unexplored territory in that area that we could look at. So that's moving beyond pharmacology.
A lot of fascinating stuff to look at for sure.
Yeah, it keeps us in a job.
Very true. Tell people where they can find and follow your work.
Oh God, I have just my website on the university, I'm not, it's funny how Twitter died, right?
How what?
Twitter used to be quite a nice place for academic discussions of psychedelics, and then it went nasty.
Not anymore, huh?
And then Blue Sky, people tried to go to Blue Sky, but it's boring. So I don't post on those social media things anymore because it's not very.
Yeah, I feel you.
Yeah, there's a gap in that. There used to be really entertaining psychedelic discussions on Twitter a couple of years ago. There was a lot of engagement, but that all kind of died when Elon Musk took over the platform. So,
too bad.
Yeah, it is a bit of a shame because there were some interesting things being discussed there. So
this sounds like an opportunity. Someone should start a social media. Website.
It was interesting. Well, people went to Blue Sky, and
Yeah.
all the people turned up, but the algorithm is so bad
Yeah.
that nothing interesting happens.
Yeah.
And so I went on to Blue Sky for a while, and then I was like, it's dead. So I was like, there's nothing interesting happening here.
Yeah.
The algorithm wasn't juiced enough,
Yeah, I hear you.
Yeah.
So basically, people should Google your name to find your research.
If you can spell it.
If you can spell it.
Yeah. I try to avoid self-promotion, so.
Got it. Well, Professor Suresh, thank you so much for sitting down and talking to me.
You're welcome, Yoshi. Thank you very much.
Much appreciated. And thanks to everyone for tuning in. My name is Yoshi from the Borealis Psychedelic Science Summit in Stockholm. Catch you next time.